actually start recording. So now I'm actually doing that. So I guess there's a little disadvantage for the other people who are not here for some reminders. But anyway, let me just go away. All right, so today we're going to talk about the biochemical process that is directly involved in generating energies for the cellular systems. And the oxidation phosphorylation is a critical biological process for such thing. And that's basically the process of electron transfer from reduced coenzymes to molecular oxygen. And reduced coenzymes, we talked about coenzymes a lot, right, when we're talking about enzymes. But particularly for this relevance, it's NAD plus or the NADH, or FAD or FAD-H2, and so on, those kinds of coenzymes who are working as an electron carrier and handing on to the oxygen molecule eventually to lead to the ATP synthesis. And for this oxidative phosphorylation, as its name refers, it requires oxygen molecule and electron transport chain basically to get the electrons from the metabolites, basically, the starting material, to hand it on to the oxygen to lead to the reduction of oxygen. Again, we're going to revisit about the reduction and oxidation a little later on, but it's all about the reduction of molecular oxygens to generate the energy. And that happens in the mitochondria. And we talked a little bit about mitochondrial structure here, but we're going to revisit that too in the next slide. But this oxidative phosphorylation happens through this using this mechanism, which involves a proton pumping. So it pumps the protons out of the mitochondrial matrix, which is the internal stuff, the liquid or gel-like stuff, into the inner membrane space, which is in between two membranes. So we talked about the structure again here, where the mitochondria have two membranes, outer membrane and inner membrane, and in between there's a inner membrane space, which is right here, and inside of that there's a matrix. And this inner membrane especially make a lot of folds-like structure like that, which touches the matrix, and it's called the cristae, basically all the folds that happens on the inner membrane. And that the outer membrane can interact with the other organelles in the cell, like a rough ER or the nuclear envelope and so on. And this mechanism of the pumping out the proton from the matrix to the inner membrane space leads to the creation of electrochemical gradient of a proton, right? Because it's pumping out the proton across the inner membrane. So you're going to have more positive charges deposited on the inner membrane space, as opposed to it's going to be stay relatively negative in matrix. And that'll lead to the ATP synthesis by powering it. That is from using this gradient. So let's look at the mitochondrial structure and the more facts there. And before we get to that, I actually wanted to mention the difference between this oxidative phosphorylation. And we also talked about substrate level phosphorylation, if you can recall, when we were talking about the glycolysis, basically, where it generates the ATP. And there are other processes too, where the different type of phosphorylation is involved. Both of the phosphorylation, oxidative phosphorylation, and substrate level phosphorylation ended up generating ATP. But oxidative phosphorylation is about using the electron transport through the electron transport chain to where it uses the oxygen, reduction of the oxygens to lead to the ATP synthesis. As opposed to substrate level phosphorylation is using the substrate itself, actually, that contains a phosphoryl group. So it used that phosphate to take it off from the substrate and then put it on the ADP to generate ATP. So those are separate processes, but ended up having the same product as ATP. So you should be able to distinguish these two different phosphorylation. Okay, so mitochondrion is a singular, and for the plural form. It's a mitochondria, and it's basically when you look at just a single blob of the mitochondrion, it has the ellipsoidal structure with the size of 0.5 by 1.0-ish, micron. So it's fairly small. It's much smaller than a nucleus. And there are typically like 2,000 or so of these mitochondria exist. And they're not just an individual, like a separate kind of organelles, and they can go through the fusion and fission and things like that, sometimes make them the huge blob, or sometimes make them the really small fragments and things like that, depending on their cell cycle stages, as well as some kind of environmental like stress or cues for the development and so on. So it's pretty dynamic organelle, but it's mainly responsible for generating ATP. So again, like we said, it has the two membranes, which is a smooth outer membrane and inner membrane. Smooth outer membrane has relatively low composition of the protein, so 60% of the protein and the 40% of the lipids. So that's why it's quite smooth. As you can imagine, membrane, the lipid bilayer is pretty smooth, right? And it's permeable for most of the things, which is smaller than 10 kilodalton. So it can go through the ions for sure, as well as the small like molecules, water molecules for sure, methanes and stuff like that. And it contains porins, which work as channels. So it has like literally like little pores that's stuck on the outer membrane. So the relatively bigger molecules can go through that pretty freely by diffusion. As opposed to outer membrane, the inner membrane, it has the much more complex in its composition, and it has a higher percentage of the protein, so 75 to 80%. So that's why it's relatively rough. There are a lot of sort of like convex that can be generated on the membrane, and this is where the ETC, electron transport chain, and the oxidative phosphorylation proteins are organized, and they basically exist there in the multi-protein complexes. And it's about, again, generating ATP, the energy. And it is extremely folded. We talked about the cristae, and it forms that really complex structures there to get the large surface area that is touching between the matrix and inner membrane, because again there are a lot of proteins that are involved in generating energies. So for the higher efficiency, you might want to have the higher surface area there, even in the small volume of the mitochondria. And this inner membrane is impermeable as opposed to permeable outer membrane. Except for the small molecules like the molecular oxygen, molecular carbon dioxide, or the water molecules. And this means that being impermeable for the most of the molecules, meaning that there should be an elaborate transport system exist for transporting some big ions or charged ions, substrates, coenzymes, and so on. And it has matrix, which is right inside of the mitochondria, which is a highly concentrated gel-like, liquid-like substance. And it contains lots of water, and as well as proteins, soluble proteins. There are mostly enzymes for generating energies, again, processing the TCA cycle and so on, as well as cofactors and different ions. And again, TCA cycle enzymes are in matrix. We talked about this, right? So glycolysis happens in the cytoplasm, but the TCA cycle happens inside the mitochondria, especially in the matrix. Except for the succinate dehydrogenase, which is bound to the inner membrane that we're going to talk about again later in this lecture. And mitochondria have their own genetic system, meaning that they contain DNA too. It's called mitochondrial DNA. What would that mean? The fact that this one has DNA, does that mean anything? Does that tell you anything? Have you heard of like mitochondrial DNA? Yes? What would that mean then? Is that important for mitochondria? maybe. Exactly. So it can tell you the origin of that. So it's probably the symbiosis that happens early of this, life form. But that doesn't mean that the mitochondria is autonomous, it actually says, fully autonomous, or it can just survive by itself because it uses a lot of proteins that are imported from the cytoplasm, which was basically encoded by the DNA in the nucleus. So there's a nice system where they developed it. Like at the beginning, they probably were quite autonomous, but ended up just evolved in a way that depending on each other, nucleus, nuclear DNA and the mitochondrial DNA. All right, but there's a transportation issue here because, like I told you, outer membrane is pretty like permeable, so everything can just go through that. But the inner membrane is impermeable, like I told you. There are a lot of proteins there, and the lots of proteins, especially involved in this electron transport chain or so. But when you think of the coenzyme like NADH, which is reducing equivalent, so NADH is a reduced form of NAD plus, right? Because it gained 2 electrons from the other metabolites basically, but that has to get into the mitochondria to fed into the electron transport chain to pass on the electrons for generating ATP in the end. But NADH is a bulky, really huge molecule, so it cannot just go through the inner membrane by diffusion at all. Much, much bigger than these small molecules. So there's got to be a system to actually transport it from outside of the matrix to the inside, going through the inner membrane, right? Because NADH is the product of glycolysis, which happens in the cytosol. It's not in the mitochondria, so it must be transported in. So it uses this malate shuttle system where oxaloacetate, again, this is the starting material or the starting molecule of the TCA cycle, if you recall it. And it reacts with the acetyl-CoA to get the acetyl group, right? And then becomes the citrate and so on. So this oxaloacetate in the cytosol, which is part of the TCA cycle, the beginning material, is reduced into malate. So it goes back to L-malate. So if you revisit the TCA cycle, this is the step 9 or something like that of TCA cycle, the last step, where L-malate is basically oxidated to become the oxaloacetate. But it has to be reduced back to malate with this using the NADH. That is going to be oxidated, right? So it's a coupled reaction where NADH is oxidized, but the oxaloacetate is reduced. And then this malate can be transported into the mitochondrial matrix, and then it's going to be reoxidized into oxaloacetate, meaning that now NAD plus inside the cytosol matrix or the cytosol of the mitochondria is reduced to the NADH. Does that make sense? So it's not about transporting the NADH molecule itself across the inner membrane, but it's about transporting the malate to change the status, the oxidation reduction status of NAD plus or NADH, which already exists inside the matrix, in the matrix or the outside in the cytoplasm. Okay, it's not just NADH, but it also have to get in the ADP to generate ATP, and also already generated ATP has to be pumped out of the mitochondria, right? And this process is done by this adenine nucleotide translocator or ATP-ADP translocator. And that basically results in the export of a charge, because when you think about the structure of ATP, which is right here, it has 4 minuses, as you can see. So it's there's a 4 negative, 4 minus that gets out, because ATP, generated ATP should be exported out to the cytoplasm or the cytosol, so it can be used as an energy source for any kinds of biochemical reactions, as opposed to ADP, which gets imported into the matrix. Well, missing this just one phosphate, so it'll have only 3 minus, right? So 4 minus gets out, but 3 minus gets in. So the net export, it's going to be the net export of 1 negative charge per ATP. And that process is going to be driven by the electrical difference in and out of the inner membrane because of the protein pumping. So like I told you, there's going to be proton pumping happening that gets the proton out of the matrix into the inner membrane. So you're going to naturally set up the more negative charge inside the matrix, in the matrix. And since ATP, ADP process is about exporting negative, you already have a lot of negative in the matrix. So it's more like a natural or favorable process that can occur, right? So that means this ADP importing and ATP exporting won't require too much energy. It's a sort of a natural process. It's following the electrochemical gradient. Any questions so far? Okay. So we're going to quickly revisit the redox reaction here. Again, oxidation reduction reaction. Nothing special. It's just quickly revisiting that. But this redox reaction is a key for the understanding how electron transport chain system works and how it is driving the setting up the proton gradient for ATP synthesis. Basically, you need two components, electron donor and electron acceptor. So it's going to be two coupled reactions, right? And the direction of electron transfer will be from the donor, which is on reduced partner. So it's a reduced partner, meaning that it's reduced. So it already has gotten electrons. So it contains electrons. And acceptors will be accepting the electron, so they don't have electron at the moment, so it's an oxidized form, so oxidized partner. It's going to be acceptor, and they're going to be basically working as reducing agent and oxidizing agent, respectively. As you can see here, we already talked about this, as you know. And coupled the process will be basically reduce the partner, which is a donor, gets oxidized, meaning that they lose electrons through the oxidation, and oxidize the partners, which is an acceptor, gets reduced through reduction. And again, these are the half reactions, but they must happen at the same time, right? And it's you can see the conversion of species here. And let's understand a little better with this redox reactions by considering the energy changes and the thermodynamics here. By understanding the fundamental properties of a reacting species in the redox reaction, again coupled to reactions, you can understand the likelihood. You can basically tell or determine the likelihood, whether that electron transfer will take place or not. And also, you can determine the direction of the electron transfer too, because one of those two reactions that are coupled should be have more propensity to be reduced, and the other should be more likely to oxidized, and they should be coupled to pass on the electrons. So to do so, you can actually look at the reduction potential, which is indicated as this cursive E. And that this reduction potential, as its name refers, tells the tendency of a given reacting species to gain electron. Gaining electron, again, it's about reduction, right? So that's why it's reduction tendency or potential. And that happens while the other partner gives up the electron. So it's that partner is going through the oxidation. And you can see these numbers that gets positive or negative and things like that on this table. And negative E reduction potential means that it has tendency to give up the electrons when paired with a certain like standard, certain reaction there. And again, giving up the electron is it reduction or is it oxidation? Giving up the electron. It's an oxidation, right? So negative value for reduction potential, it kind of makes sense, right? You have a negative value for reduction potential, meaning that you're going to likely get more oxidation. As opposed to if you have a positive E, reduction potential, of course, you have a higher potential for reduction. So that reaction is going to get, reduced, meaning that it'll attract and gain the electrons when it's paired with the other, the half reaction. And there is a standard reduction potential, which is written as cursive E naught. And that is basically reduction potential that can be measured at the standard state, which is 25 degrees C, 1 atmosphere, and all the reactants and the products at the 1 molar concentration, basically. And this is the most of the reactions is already measured and known for this standard reaction potential E naught. And here's a table for that, and you can see a lot of them. And all of them are written in a way that the reaction is reduced, even if it has a tendency to be oxidized. But still, since this is a reduction potential in convention, everything's written that way. So you can see that all the reactions contain electrons here on the left side, and basically it gains electrons, so it's reduced to become something else, right? And all the reactions also contain hydrogen cation, meaning that this hydrogen cation reaction that is reduced or oxidized basically is a coupled reaction and is used as a standard. Here, how can we actually measure this reduction potential, E? We can measure it empirically by using this voltmeter and just connecting those two half reactions and reading the electrical changes, basically current there that is set up. So there must be two half cells which contain two half reactions, basically. So that's redox couples. And one contains the sample half cell that you're going to measure for its reduction potential. So it contains it can be either reduced or oxidized forms, depending on what kind of things you're measuring. But it has to be at 1 molar because you're measuring the standard reduction potential. And the other partner, the other half cell, should be a standard. So it should be the same thing over and over again, even if you're changing the sample half cell. So you can get the like all the values relative to the with the same like standard or the same starting point. So you can make the fair comparison between the other samples. So this standard half reaction contains again hydrogen cation. At 1 molar, and also the hydrogen gas at 1 atm. Again, everything is at the standard state. And this voltmeter reports the direction and magnitude of electron flow, which is driven by the electromotive force. Depending on where the reduction happens, it'll actually gets the attract the electrons, right? So the oxidized reaction should give up the electrons, so it's going to go through the voltmeter and will hit it to give the certain value there. So if the electron moves towards the standard half cell, which contains this hydrogen atoms and ions, then it means that the reactant that you're setting up, the sample, gets oxidized. Does that make sense? Because the electron is moved away from your reactant to your standard half cell. So it is basically leaving your reactants. So you're losing electron, and that means it's oxidized. And being oxidized meaning that you're going to have a negative E reduction potential. And if the electron moves away from this standard towards your reactants, that means your reactants are gaining the electron, and that means they get reduced, and that means it has the positive E value. Okay? All right. So like I told you, this is just a simple measure where you can get the reduction potential. And using the same standard over and over again, you can get this standard reduction potential, and it has to be happen at the standard state where 25 degrees C, and everything's at 1 molar. But if the reactant is at pH 7, then it's going to be called the biochemical standard reduction potential, because now we're using biochemical standard state instead of just a chemical standard state, right? And it's written as this E naught prime. And you probably can remember, when we talk about delta G, we talked about delta G naught, which is at the standard state for the free energy. And delta G naught prime is basically for the delta G value under the biochemical standard conditions. So same thing applies to this reduction potential E too. And again, it measures the potential or the tendency of this chemical that is rather reduced compared to the standard. And again, it's coupled with the hydrogen. So that means the hydrogen atom, the reduction of the hydrogen atom is set at delta E, E naught as a 0 volt, because it's just a standard. And again, same thing applies to the delta E naught too, as we used for just E values. Again, E naught is a standard reduction potential, and just E is a reduction potential, so it's a relative value. And when it's positive, that means reactants is reduced. When it's negative, that means the reactant tends to be oxidized. And when you look at the table here, the top one here is the reduction of molecular oxygen, as you can see here, with the value of E naught prime as 0.815, and that's the highest value. And this electron flow basically depends on the what kind of partners you have. So that means even if you use the same half reaction, that can be either work as a donor or acceptor, depending on your partner where that is sitting on this table. So let's take an example of these two half reactions, how the redox reactions happens, and how we can understand in terms of thermodynamics and the reduction potential. So I took out this reaction from the as you can sort of remember, this is in the TCA cycle, where the isocitrate is going to be oxidized to become alpha ketoglutarate. And here's a reaction, and the NADH is NAD plus is involved there, working as an oxidizing agent. So it itself gets reduced to NADH. So the half reaction would be this NAD plus gets 2 electrons, and there's a 2 hydrogen cations there to get the NADH and H plus. And by convention, even if the other reaction just happens like in the opposite direction of the reduction, like oxidized, we still write everything in the form of reduction for their reaction. So alpha ketoglutarate goes back to get reduced, to get the 2 electrons, and carbon dioxide is involved here, which was decarboxylated on the this intermediate state, which is oxalosuccinate, and gets back to the isocitrate. And these are the 2 E naught values, standard reduction potential values. And both of them are negative. So that means they're going to get reduced or oxidized compared to the standard, which is reduction of the hydrogen cations. Is it going to tend to be more likely reduced or oxidized when it's coupled to the standard? It's a negative value. It's going to be oxidized because it's a negative value. Again, positive value for the reduction potential, meaning that it's going to get reduced more likely. So it's going to be oxidized. Even if both of them are going to be oxidized, if it is coupled with the standard, if these two reactions are coupled, then now you need to look at the relative negativity or the positivity for these reduction potential values. So how you look at it is consistent, where the more negative value will likely be more oxidized. So here, minus negative 0.38 volt is a little bit more negative, right? So this is going to be more oxidized when it's coupled to the this NAD plus reduction. Does that make sense? So electron always spontaneously flow from the half reaction with the more negative, which is tend to be more oxidized, to the more positive one, which is going to be more reduced, gaining electrons. But that doesn't mean that the opposite thing's not happening at all. It can happen, depending on the temperatures or the concentration of the reactants and so on, and also just by chance by like Brownian motions and things like that. But typically, when you have this opposite reaction happens, you need to input an energy, so it's going to be an endergonic process. So by looking at this standard reduction potentials, you can tell that isocitrate is a donor. This one is a donor, so it's going to get oxidized. And the second half of reaction, this reaction goes the opposite way, the reverse reaction. So it's going to oxidize as opposed to this NAD plus is going to still be reduced. Okay, we're still talking about the same reaction here, where isocitrate is oxidized to become alpha ketoglutarate. But now let's think about this delta E naught value more closely. We can actually get their differences, and it's called the standard reduction potential difference, where you subtract the donor the E naught value from donor from E naught value of the acceptor. And when you apply those numbers there, you get the positive value, 0.06 volt. And there's a equation that relates this delta E naught to delta G naught. So you can relate the standard reduction potential difference to the free energy. So you can kind of tell that if this reaction will likely occur spontaneously or not by looking at the delta E naught value. So that equation has delta G naught equals negative NF delta E naught. And here N is the number of electrons that is transferred for those redox reaction. And F is a Faraday constant, which is written right here. And again, positive delta E naught, meaning that it's going to get reduced, right? More reduced. And that means, this positive delta E naught value actually, regardless of reduction or the oxidation, will result in negative delta G naught value when you think about the relationship between those two state functions, right? Because there's a minus here. So positive delta E naught will lead to negative delta G naught, and that means you're going to have a spontaneous reaction, that forward direction. And let's plug in those numbers again. So you got 0.06 volts for the here delta E naught. So you put it in there and also put the Faraday constant. And there were 2 electrons that were moved from isocitrate to NAD plus. So you put 2 here, ended up getting negative 11.58 kilojoule per mole. So it's a negative value for delta G naught, meaning that it's going to be a spontaneous process. And here's a little warning that some reactions can happen in the opposite direction to that is suggested by this consideration of uh, delta E naught value. And that is because some reactions can happen with other reactions involved too. So even if there are two, redox half reactions, there are other reactions. For example, here's a decarboxylation, is one example here. Other reactions are coupled there too to make things a little bit more complicated. Also, these reactions can happen under conditions other than standard conditions. So it can also change the direction or the spontaneity, affect the spontaneity of the reaction. So there are a bunch more other stuff here related to thermodynamics. But like we just said, even if delta E naught tells something like a spontaneity, sometimes it doesn't happen because it doesn't consider the actual condition in the cell especially. So Nernst equation considers that condition where it really happens inside the cell or some other conditions other than standard state to get the true delta E naught, and it's called the delta E naught prime. Again, it's a biochemical standard state. So here, the delta E naught values can change, depending on the concentration, temperature, and so on. So Nernst equation gets the delta E naught value here, but also consider the other energy that can be induced by the conditions other than standard state. So that's going to be just using a simple, energy equation here, RT divided by N Faraday constant, NF, and that's going to be multiplied by Ln and the ratio of the concentration of products and reactants. And you're pretty much familiar with this kind of concept here, RT, Ln, and KEQ or things like that. It all talks about the energy, right? Delta G value. So that value, the energy value, the equation is changing or modulating this delta E naught value to make it, into put it into the right context. So simply we use the same equation here, but now change it to delta G naught prime instead of just delta G naught. It's going to be same thing, negative N F and then delta G naught prime. So let's consider the one reaction, the redox reaction here. The oxidized A, because it's oxidized, they lost electrons, so it's positively charged. React with the reduced form of B, which does not have charge. Then the reduced one of the A will now gains the electron, so it's going to be with no charge, and the B oxidized form will have charge, positive charge. You just plug in these numbers because there were 2 electrons moved. You put 2 on this N, and you keep the Faraday, and then put the concentration of a product on the denominator. So these 2 concentrations, and put the other concentrations on the denominator, and then basically put this value into the this equation to get the delta G naught prime. And again, you need to put the 2 for this N. Okay. Any questions so far? All right. So we just talked about this kind of energy changes and the getting the delta G value from the delta E value, which is reduction potential, and so on. Now we want to look at it how it is actually applied and how it is relevant to our actual process that happens in the electron transport chain in the mitochondria for generating energies. So again, it's about getting the NA, using the NADH and FADH, which are coenzymes, and transfer electrons to ultimately to the electron acceptor, which is molecular oxygen in the mitochondria. So it leads to the reduction of oxygen again. And energy that is obtained through this redox reaction, it involves lots of different steps in the, again, electron transport chain. It's going to be used for driving ATP synthesis. But indirectly. It's not going to generate ATP by itself or directly. It's just generating the setting up the proton gradient, and that proton gradient will be used for generating ATP. And again, we're going to look at that process too. But the ultimate reaction of this oxidation of NADH would be right here. So NADH gets basically reacts to the half molecule of the molecular oxygen to generate this one water molecule. When you split it into two half reactions, that's going to be here the reduction of NAD plus, and also it can be here's the reduction of the oxygen molecule, the half molecule here, and you can see the reduction potential values here. One's obviously negative, and the other's positive value. And again, positive value meaning that it's going to tend to be more reduced, right? So this reaction is going to go forward as opposed to this reaction will go backward. So what the delta E naught prime will be, again, we use this relationship to get the difference. So we put the acceptor, which happens, the reduction, reaction, which is within this potential value. So we put 0.815 here, minus, and here negative value to get 1.13 volts, which is a positive value for delta E naught prime. And we plug this number into this equation to get the negative value of delta G naught with the really highly negative value, 219 kilojoule per mole, and meaning that it's spontaneous reaction. And also, it actually releases a lot of energy during this reaction, right? So this amount of energy is sufficient to drive synthesizing about 7 ATP, because when you consider the synthesizing 1 ATP molecule that costs 30.5 kilojoule per mole at the standard state, and it goes a little bit up in the cell, like 40 or 50 kilojoule per mole that we, as we discussed previously. So simply just 219 divided by 30.5 will get the 7 or so. And but when what it's really happening is that not just this one reaction happens in the one place, but it happens through the series of like other reactions, so step by step. So it actually loses some of the energies in the form of heat or other things, other forms. So ended up just having only energy to generate about 3 ATP or so, specifically 2.5 actually, from the one NADH that is oxidized. Question. Yes. So it's just by convention to put all the reactions, half reactions in the form of reduction for sure. And this is an empirical value. Actually, you measure this value, and it's happened to be negative, meaning that it's likely oxidized. So that is correct. When you look at the full reaction, that the first reaction should be oxidized, so goes backward. Any other questions? Okay, so we covered quite a lot of things so far about how to get the reduction potential, how we can interpret it in terms of the spontaneity of the reaction, and so on, and how it is applied to our mitochondrial system, where the electron transport chain works for handing on the electrons for series of reductions, basically getting on the electrons, and then ended up just generating energy. Let's look at how it is really happening in the mitochondria, especially on the inner membrane. So there are two pathways of the electron transfer. The first one is starting from NADH, and NADH is the product of a glycolysis, as you know, as well as TCA cycle, you get NADH. It is important to remember how many molecules of NADH and FADH are generated. So NADH is here, and that gets into the complex 1 of the electron transport chain. And then it basically hands on the electrons, and then those electrons move to the coenzyme Q here, and then onto the complex 3, and then also move to the cytochrome C and complex 4 eventually, where the molecular oxygen is reduced. There's another path that starts from the FADH. Again, this is another coenzyme, right, for the oxidation of the glycolysis. But it bypasses the complex 1, but gets to the complex 2 directly. And then coenzyme Q, and pretty much the same. So again, NADH gets to the complex 1, but FADH2 gets to complex 2. That's the main difference. The rest of them are the same. So now let's take a look at each of complex, and see what happens there in terms of electron transfer. So for the complex 1, since it uses NADH, it's called NADH coenzyme Q reductase, also known as NADH dehydrogenase, and it kind of makes sense, right? It takes up the H from NADH, so dehydrogenase, and it's reducing it, so it's reductase for coenzyme Q here, the next path, and NADH, which is a starting point. And this is a membrane-bound, large multi-protein complex, and it has more than 30 polypeptides, so it's a huge protein complex. And it also contains FMN, which is a flavin mononucleotide, and 6 or 7 iron sulfur clusters too. And this is a critical one for the transferring electrons because it can work as electron carriers. It has the iron ion, which can change the valency from 3 plus to 2 plus, and of course, vice versa, 2 plus to 3 plus 2. So it can hold the one electron and just to give it up to the other one on the downstream of the process. And what complex 1 does is transfer the electron pair. So it's 2 electrons from NADH to coenzyme Q, and this coenzyme Q has different other names like ubiquinone or the UQ. And this is the full reaction where redox happens, oxidation and reduction happens. NADH, coenzyme Q, which is oxidized form. Oxidized meaning that it does not have electrons, right? And then NADH basically gives the electron to coenzyme Q, so it's going to be reduced form, and NAD will be oxidized to the NAD plus. And delta G naught value is negative 69.5. So is it spontaneous or non-spontaneous? Spontaneous. I'm pretty sure you guys know the answer. So, and since delta G naught has positive value, delta E naught prime will have a positive value, right? Because there's a relationship from the equation. And path of electron is basically starting from NADH, that hence the 2 electrons to FMN here. And there are some energies that are involved for, basically like, how do you say, outputting the, generating energy, or the sort of outputs the energy through these processes are written here. So electron is transferred from FMN and then to the iron sulfur cluster, which have like 6 or 7 iron sulfur little clusters here, and then ended up handing on to the coenzyme Q, which is UQ right here. And this coenzyme Q, which is drawn a separate thing from the complex 1, is a mobile carrier. So it can just diffuse on the membrane here and there, and it just happened to just meet and interact with the complex 1, where complex 1 already contains electrons, so it can naturally hand it on to the coenzyme Q, and then the next step can occur after that. We're still talking about the complex 1, but it is quite critical because it is switching its mode for interacting or holding the electron from um, like harboring two electrons to just a single electron, and that is mediated by this FMN, the first one, that gets the electron from the NADH, which can have the either one electron or two electrons. But NADH is exclusively containing two electrons because NAD plus can accept one electron, and H can be considered as a hydride, which is a negative charged hydrogen ion, can also get the one more electron. So it's going to be two electrons. So here's the reaction again. Gets the two electrons here. But the iron sulfur cluster can have exclusively just one electron. So there's a difference here. Discrepancy happens where starting with the two electron, but have to just hand one electron. So this FMN is really useful here, where you can hold the two electrons and release just one by one to the other later system, the pathway on this complex 1. So it basically serves as a link between this NADH and the iron ion. And complex 1 provides a sufficient delta G naught, we just looked at it, the value, to catalyze the production of ATP molecule. But again, this does not happen directly. It's nothing to do with the binding ADP and just putting the phosphate there to make it into ATP. It's only about setting up the proton gradient across the inner membrane. So it pumps out the protons, which is H plus, hydrogen cation, from the matrix to the inner membrane space. And extra energy can be also released in the form of heat. When you look at the stoichiometry of the how many protons are pumped out based on the feed of the electrons, there are 4 protons that is transported out of matrix for each for 2 electrons. So like I told you, NAD plus gives 2 electrons, and the FMN basically kind of modulate its flow, but in the end, it's gonna actually go through the both electrons, and ended up having 4 protons that is pumped out of the matrix. And this 4 proton goes from matrix, which is called N side for negatively charged side, to the inner membrane space, which is called P side, or positively charged side. And there are well-known inhibitors for this complex 1. These are 2 examples. First one is rotenone, which is a plant toxin that is used by the Amazon tribes to basically catch the fish by poisoning fish neuronal system. And the other one is amytol, which is a barbiturate derivative, and it is used as a pre-anesthetic agent or sleep disorder drug. Now we're going to move on to the next complex, still within the electron transport chain, complex 2. And that complex 2 introduces electron from FADH2, not NADH, right? It's a separate process, into the electron transport chain, especially to coenzyme Q. It's also known as a succinate coenzyme Q oxidoreductase. So it is involved in redox reaction, and it links the succinate and coenzyme Q. Here's a succinate that gives the electrons and becomes a fumarate, and it gives the electron to FAD to make it FADH2, which is already included in complex 2. Okay. It's not actually getting the FADH2. It already has FAD there, but the FAD is reduced from the succinate to gain the electrons. So it also contains a succinate dehydrogenase to get rid of hydrogen electrons from that. And this is the only enzyme of TCA cycle that is membrane bound. So just Remember all the TCA cycle happens in the matrix of mitochondria, and all of the enzymes are soluble, so just located in the matrix, except this succinate dehydrogenase. So you can revisit the TCA cycle and see that, and that's a nice link between TCA cycle and the ETC, electron transport chain. So here's the reaction, the total reaction with the delta G naught prime as a negative value here, barely negative here. And delta E naught prime is, of course, a positive value, 0.03 volts. So that means it's going to happen, spontaneously again. And it has also the large protein complex with the 4 subunits, as you can see here. 2 of them are responsible for anchoring into the inner membrane of mitochondria, and rest of them are basically exposed to the matrix to interact with the succinate to get the electrons. And it also contains, similarly to the complex 1, contains the iron sulfur cluster, but only 3. And also there's again bound the FAD there. So the electron starts from succinate, gets to the FADH2, and then that hands on the iron sulfur cluster, and eventually goes to the coenzyme Q for the later step on the complex 3 and 4. And it does not release enough energy to synthesize ATP because it does not set the proton gradient at all. So when you look at it in this cartoon, the complex 1 pumps out the 4 protons, as opposed to there's nothing written here for the complex 2, right? And it really doesn't happen. So it does not, it's not involved in the proton pumping, meaning that it actually does not produce any energy for ATP synthesis. But it only serves as the injecting or handing on the electrons to the later stage of the electron transport chain, which is coenzyme Q. So that's a big difference between complex 1 and complex 2, or using the NADH versus FADH2. So NADH is better in a way that it generates more energy because it sets up the protein gradient, as opposed to FADH2 is not involved in protein, setting up the proton gradient. So it's not going to provide much energy as NADH does. Okay, so the next step is coenzyme Q. Again, it's also called ubiquinone, or UQ, or the UQH2 when it's reduced. And it's a mobile electron carrier, accepts either 1 or 2 electrons, and it has hydrophobic tail. Its structure is a pretty simple molecule, where it has a phenyl ring, as you can see here. And then it has a long chain, hydrocarbon chain, which serves as a tail. And basically this tail is stuck on the inner membrane. And it can diffuse freely in the inner membrane, basically, to interact with the complex 1, complex 2, as well as complex 3, and works as a shuttle, basically, for the electrons. And here's a complex 3, and it's also known as coenzyme Q cytochrome C reductase. Again, it links the CoQ here, coenzyme Q, and the cytochrome C for handing on the electron. And it hands on the electrons from these 2, and with this net reaction here, delta G naught value is still negative, with the delta E naught prime value positive. Did something bad happen, or hopefully not? Okay, I'll just ignore that for now. Hopefully we don't need to be evacuated. So, but it's a part of the complex 3 is still part of the inner membrane. So it's stuck in the inner membrane. Contains 2 type B cytochromes, where you can see it here, cytochrome BL and cytochrome BH, and also 1 cytochrome C1 and 1 iron sulfur cluster here. And here, cytochrome BL means that it has low E naught value, and H, of course, has the high E naught value. And the path, electron path is complex because as you can see, it goes back and forth between coenzyme Q and complex 3, like, coming back and forth. But this system is called the Q cycle. And it also provides a sufficient energy to synthesize ATP molecule because as you can see, there are 4 protons that are pumped out from matrix. Again, contributing the setting up the proton gradient. But it interestingly acts as an unbalanced proton pump. So when it happens, it actually takes up the 2 protons into the matrix, but it outputs the 4 protons out of the matrix to the inner membrane. So there's a unbalance basically here, and this unbalance or disbalance is corrected by the next step happens on the complex 4, which is unbalanced in the opposite way. So it's gonna just cancel out this difference. And a potent inhibitor for complex 3 is antimycin A, which is a pretty well-known antibiotics. And the next one after the complex 3 is a cytochrome C. And that's the only electron transport chain components that is water soluble. So it is water soluble. So it looks like a small globular protein, and it resides in the inner membrane space. It's loosely associated with the inner membrane and has a porphyrin ring, which structure you can see right here. And this porphyrin ring can harbor basically positively charged metal ions right in the middle there. So that metal ion is important for holding the electron, something similar to the iron sulfur cluster. So it can be iron ion that is sitting right here. And it serves as a mobile electron carrier and moves along the surface of a membrane basically by diffusion, interact with the complex 3 as well as complex 4, and exclusively carry 1 electron there. All right, so the next one, the last one in the ETC is the complex 4, also known as cytochrome C oxidase. It oxidizes cytochrome C. And it accepts the electrons from specifically 1 electron, right, from the cytochrome C, and delivers them to eventually to the molecular oxygen. And here's the reaction. If it gets the 2 electrons, then that's going to be the total reaction, redox reaction, where 2 cytochrome C is involved because again, cytochrome C can carry only 1 electron. And then there's a 2, hydrogen cation, and the half molecule of the oxygen here, and there's oxidation of the cytochrome C and the reduction of this oxygen molecule to get the water molecule with the delta G naught prime value highly negative. So and of course delta G E naught prime will be positive value, and that means it releases a lot of energy when it happens, and it's also spontaneous process. And again, it depends on the inner membrane composed of at least 10 subunits. So it's a pretty bulky complex too, including cytochrome. So you can see all these here: cytochrome A, cytochrome A3, and 2 copper ions, copper A and copper B here. And these copper ions are cycling through this changing valency too. That is for the electron carrier. Transferring the electrons. And again, this one is also just 1 electron carrier. And here's the path that the electron goes to the cytochrome C first, cytochrome C to copper A first, and then cytochrome A, and then it gets to this little complex here, but first handed on to the copper B and then cytochrome A3, and then eventually gets to the oxygen molecule here. And as you can see here in the delta G naught prime value, it generates enough energy to synthesize ATP molecule, and also again acts as an unbalanced proton pump, but in the opposite way as the complex 3 does. So 4 hydrogen cation or the proton gets into the matrix as opposed to only 2 hydrogen cation is or the proton is exported out to the inner membrane there. The inhibitors are the cyanides, azide, carbon monoxide, and so on. Now we talked about the electron transport chain there. It's all about handing on the electrons and setting up the proton gradient, right? There's nothing to do with ATP synthesis at all. Now we need to make connection with this electron transfer to the ATP synthesis, and how the ATP synthesis actually happens and how it is powered, meaning that where it gets the energy from. Like I told you multiple times, it's from the proton gradient that is set up across the inner membrane of mitochondria. But let's just think it logically here. Electron transport is exergonic, meaning that it generates a lot of energies, and the energy should be collected in a certain form to use it for ATP synthesis. But no high energy intermediates are used there. And if you recall, the high energy intermediates contains a phosphoryl group, phosphate, basically, that can work as a substrate level phosphorylation, but there's no such thing happens. So that's why it's not the substrate level phosphorylation, but the oxidative phosphorylation that actually drives the ATP synthesis. So nothing could be seen for these high energy intermediates there that provides a phosphate group. So that must be something else, right? So Peter Mitchell in 1961 proposed this hypothesis, and it's called chemoosmotic hypothesis, which was really kind of a new idea, but it obviously worked really well to explain how it is working. So he earned the Nobel Prize in 1978 for this hypothesis. And it basically says that the energy that is gained or exerted from this electron transport system or the chain will be used to create the electrochemical proton gradient across the inner mitochondrial membrane that later is going to be used for ATP synthesis. So let's just think a little bit more about how you can get the energy for ATP synthesis here. So let's get the delta G value, and delta G is important again to see that if whether this is spontaneous or not, so you can naturally get this reaction going, and also you can estimate how much energy is output, like how much energy you can get from this reaction too, if it is a spontaneous, right? So delta G for setting up the gradient here will be RT ln and the ratio of the concentration of hydrogen cation concentration, proton concentration inside the matrix and the outside of the matrix, which is the intermediate space here. And also Z is the number of electrons or the protons that are moved. There are different charges there. And Faraday constant and the delta psi is the electrical potential changes across the membrane. So it can be considered similar to the reduction potential too. So that's why this one looks quite similar, right? And there's a plus because it was initially minus from this energy here to get the delta G because it goes the reverse direction. So here, H plus concentration can be converted to pH, right? But we have to put the minus log there. So it's already inside the natural log. So changing the natural log to log of 10 with a base of 10 will require this simple like transition transformation where you can just simply multiply it by 2.303. So you get the this 2.303, and then again it should be minus log. So you put the minus negative value here, and then you're gonna get the negative log of H plus concentration. So it'll convert it into pH inside and pH outside here. And then basically this is the changes of the pH inside and outside, right? And this is the equation you can get to estimate the delta G for setting up the gradient here, not for setting up, but using the gradient here. The pH value is lower outside of the matrix because proton pumping. All the protons are exported into the inner membrane space from the matrix. So you always have more protons in the inner membrane if the inner membrane is intact, and you're going to have a higher hydrogen cation concentration, meaning that you're going to have lower pH value, right? And typical value for the delta pH, which is right here, pH in minus pH out, is about 1 here. So we can use number 1 here. And delta psi is around 0.18, so we can actually put in here too. And rest of the other values are already known. The gas constant, temperature, which is 37 in the cell. You can derive the delta G here, which is 23.3 kilojoule per mole. And that means protein transport from matrix to intermembrane space is an endergonic process with the positive delta G value. And that also means that the other way around, where the proton comes into the matrix, is a spontaneous process, right? With the delta G value negative 23.3 kilojoule per mole. Checking the time. So that means this system can use this delta G value to drive ATP synthesis. So let's think about how much of ATP can be generated here. At the standard state, ATP synthesis costs 30.5 kilojoule per mole. And under the biochemical standard conditions, it's going to be 40 to 50 kilojoule per mole, where all the concentration for the ATP and ADP as well as temperature is in consideration there. And that means 2 or 3 protons must be transported from across the inner membrane to get enough energy to drive ATP synthesis. So here's a 23.3 kilojoule per mole that is set up for the delta G for the proton gradient. And if you make 2 or 3 times, that's going to get around 46.6 to 69.9 or so, which will cover this ATP synthesis cost at the biochemical standard state. And this chemoosmotic theory explains pretty well of all these phenomena here, and that matches well with the empirically measured values for delta G or the delta E naught and so on. So for this theory, it requires an intact inner membrane, because when inner membrane is affected or loose or have holes and things like that, then it's not going to be able to set up that nice proton gradient. So it has to be impermeable, especially for the proton, H+, as well as some other ions that can change the charges, right? And again, the free diffusion can dissipate this electrochemical gradient. And this electron transport happens through the electron transport chain, resulting in the proton that gets out of the isolated intact mitochondria. That is the experiment, actually proven by the experiment. That they purified the mitochondria and saw that there's a proton constantly spewed out from not spewing, but exported out the mitochondria. So that was proven that this theory or the hypothesis is correct, or at least supporting this hypothesis. And again, adding some compounds or the inhibitors that increase the ion permeability, so it disrupts the structure of the inner membrane, basically, also affect the function of a mitochondria, and that particularly decouples the electron transport and the oxidative phosphorylation, where it generates ATP synthesis. So that means, again, setting up the proton gradient is important, and keeping the inner membrane intact is important. Okay, so we have like 2 minutes or so actually, but I'm going to spend 2 to 3 minutes or so to explain this system, and that's going to be it. What I'm going to explain now is the ATP synthase and the enzyme that is involved for actually using the proton gradient for driving or generating ATP. So here's a different name for that. It's widely used. F0, F1 ATPase is also widely used name for this. And it has two big components, F0 and F1. F0, as you can see here, is the component that is stuck in the membrane, inner membrane of mitochondria there, as opposed to F1 is exposed to the cytoplasm, and that means a matrix, exposed to the matrix of the mitochondria. So F0, as you can see, interact with the lipids here on the membrane is water insoluble transmembrane protein. It has three kinds of subunits with the stoichiometry. 1A, which you can see in both here, cartoon or the structure here, but there's one big A here, and 2B that looks like a big, long rods, and the 9 to 12 or the 10 to 15 C subunit, depending on what kind of species that you're looking at. And this F0 forms the transmembrane channel, basically. That's the proton that flows to the F1. And that's down the electrochemical gradient, right? So it's gonna happen like all the proton that resides in that is in the inner membrane space will just leak through the matrix through this F0 channel. And F1 is a water-soluble peripheral membrane protein that is responsible for ATP synthesis. So it has ATP synthesizing activity, and it's sufficient by itself. And it has a composition of alpha, beta, gamma, delta, and epsilon here. And there are 3 alpha and 3 beta components here. The alpha binds the ATP, and beta makes generates the ATP from ADP. So this AB complex, basically, the little like a protomer, has this catalytic site for ATP synthesis. And here the gamma forms the stalk that holds this alpha beta complex here and serve as a gate controlling of this ATP and ADP. And the delta, which is right on top of here, and the epsilon, which is interacting with the C subunits on the F0, is interacting with this F1 and holding this F1 and F0 together. So when you look at it, it has the whole like a long pole, which is B2, and then it holds the sort of a circular structure here, just like the merry-go-round kind of thing. So it actually rotates there. And how it works is that it has the 3 alpha beta protomers, which has 3 distinct sites there. So again, this is total of 6 subunits, but alpha and beta makes the 1 protomers and have the 1 open sites there. So, and it can also have the 3 different conformations or states, which is O form, L form, and T form. And I'm sorry, I have to just go because this is important for your final exam. So that's why I'm covering this. So it has the O for the open form, which is inactive form, and it cannot bind the ATP or ADP. It has L form because it's loose binding, and it's an inactive form. But the T form is for tight binding, so it's active for the ATP synthesis. So how it works is that it goes through the 3 steps here. For the first step, when you look at it, the L site, ADP and inorganic phosphate binds to L site, which is loose binding. And then previously synthesized ATP is sitting on the T site, ready to be released. And O site is basically empty, which is open. On the step 2, there's an energy that is driven, driving the simultaneous conformation of L to T, and here T to O and O to L. And upon this conformational changes, ATP is going to be synthesized on the T form, which is again for tight binding. And ATP will be released, that is already synthesized, will be released from this O, which used to be T, but it's open, so it can release it. And this upon the O to L changes, now it can have affinity to bind the ATP and inorganic phosphate, and it basically cycles through these 3 steps. And so basically L changes 2 for these 3 steps. So for the next 3 steps, T will change it to O, and then O will change it to back to L in the later 3 steps. So if it goes 2 more cycles, it's gonna go back to its original state where it is L on the top, like that. And these are the inhibitors of the ATP synthase, oligomycin B or DCCD. And what it does is that they bind to the F0 and prevent the ATP activity, and it's all about the using or not using the proton gradient right here. So it'll inhibit the using F0 going through that proton that fed to the F1 of the ATP synthase. And the last one is the ATP translocase. Let me just quickly see if there's anything important. Translocase that kind of flips around from gets the ADP in to the matrix and ATP out of the matrix. And how it works is that it binds to ADP outside the matrix, so in the inner membrane, and then flips and then releases ADP inside the matrix, and then binds the ATP, and then flips again to release the ATP to the inner membrane. And it's in the end will be make the exporting one negative charge, because it exports the ATP, which has 4 negative charge, but it imports the ADP, which has 3 negative charge. So that means you're exporting just 1 negative charge for 1 cycle, right? And that is the same as importing 1 positive charge. So exporting 1 negative or importing 1 positive is sort of the same thing. And that actually works just following the down the electrochemical gradient, because as we talked about proton pumping, the positive charge is set up outside the matrix, inside the inner membrane space there, and this one is about importing 1 positive charge into the matrix. So again, this is not against the proton gradient. It's just following this. So that means it won't require energy to do this. Let's just to quickly go that. This is quite important here. I'm going to just jump to the important point here, where in the end, here's an ATP. For generating 1 ATP, you need 2 to 3 protons that we calculated based on the delta G value. And for the translocation of 1 ATP, 1 ADP, you also getting the 1 proton into the matrix that we just talked about right here. And that means you need 4 proton movements down the gradient to get the 1 ATP. Let's think about the cost and effect here. That you basically exports out the 10 protons through the electron transport chain, but you only need 4 proton import, or just going through the down the gradient to generate 1 ATP. So 10 divided by 4, meaning that with just 1 flow of 2 electrons through the electron transport chain, you can get 2.5 ATP. Again, this is for the 2 electron that is transported. But when you think about the complex 2, it actually skips the complex 1, so it'll only pump out the 6 protons, but you still need the 4 protons that gets into the matrix to get the 1 ATP. So 6 divided by 4, so you're going to get 1.5 ATP, meaning that again, with NADH, you're going to get 2.5 ATP. But with the FADH2, you're going to get only 1.5 ATP. So that's the difference here. Uncouplers, you can just read it, basically, information. And the radicals are important too, but let's see. I'm going to just explain just one thing here, that the free radicals are bad because they are reactive. That's pretty much it, actually. And they can go through this series of reactions that happens, keep generating radicals. And these free radicals are basically having some unpaired electrons on the outer shell. So it'll induce, we'll try to get the other electron from other molecules. So it's going to get reduced, meaning that it's oxidizing other molecules. So that can be a really bad thing. So it can generate the really bad radicals, highly reactive radicals like hydroxyl radicals and so on, and can disrupt a lot of macromolecules like lipids, DNA, and proteins and so on. But it can be resolved by antioxidants like superoxide dismutase and so on, which basically uses this highly reactive radical species back to the hydrogen peroxide or the water molecule. Okay, so well, we can just skip this recap, but basically what we've learned so far is that there's a we talked about all the enzymes that are involved for making these big molecules into small molecules, and also there are glycolysis through the glycolytic pathway, and we talked about all the enzymes there and what are the products of that, especially NADH here, as well as ATP. And this is by the different process, whether you get the ATP here. So it's either going through the oxidative phosphorylation or the substrate level phosphorylation. And we talked about the citric acid cycle or TCA cycle and the ETC, basically. And it's all about generating energies. And where it happens is also important, whether inside the mitochondria or in the cytoplasm. Okay, so that's it. I'm sorry that it's just gotten so long, but next time we're gonna have the review session. You can just pack and leave, but where we're gonna have a similar format, so you already can see the practice exams in Blackboard. So try that, and we're gonna talk about those problems as well as important points for your preparing for your finals. All right, that's it.